introduction. Hello, everyone. My name is Mishelle Solis Juarez. My internship was the study of glycoprotein for vaccine discovery at the University of Stanford in the Department of Structural Biology in Professor Theodore Jadetsky's lab. I worked under Javier Najib, like Sophia mentioned. So the focus of our lab is the human cytomegalovirus, which is a common herpes virus that infects most people. HCMV can be very destructive and it causes serious harm to immunosuppressed individuals like cancer patients and babies. There is currently no effective therapies for this virus or vaccine, but potentially getting a vaccine will help babies especially because they suffer long-term defects. The CMV virus envelope is made up of many glycoproteins that are used to get inside the cell. There are quite a few of those, but we're going to actually focus on glycoprotein B, which is the one that helps the virus enter the human cells from a compact, unstable prefusion, which is this one right here, to a stable rod-shaped postfusion, which is this one right over here. And there have been many studies, research done mainly on GB, but for the most part, research has been done on postfusion GB, which is this rod shape here, because it is the most stable. And it has, unfortunately though, there has not been success in the vaccine development because we think that at this stage, it has already fused with the host membrane. So we want to focus on pre-fusion GB, the compact, but it has just been very difficult because of its instability. Right now, there is currently one full-length pre-fusion GB, which is by Pfizer, and this gives us clues, but it's not a good candidate for a vaccine because it is chemically cross-linked and not stable. And as you can see here, this longer structure where this red circle is, this is where the GB structures focus on stabilizing the region. My mentor, Dr. Najib, has actually been able to stabilize pre-fusion GB and without the cross-link, but it is still missing like the transmembrane. And the goal of my internship was to stabilize longer versions of GB in pre-fusion form so that we aren't missing any important regions of the protein itself. We began my internship by evaluating various GB mutations that were expressed in mammalian cells that were put in a Western blot dot to detect the presence of the GB protein. As you can see, these are our results. As you can see, some of these are expressing well compared to our prefusion, which is our control, which is the TRGB647 revert. The mutants were encouraging because a lot of these mutants were secreted in soup and soluble. Once this showed that some of the mutants are secreted and soluble, we did another Western blot, but this time we did it with an SDS page gel to evaluate that the secreted proteins were the expected size, which is 200 kilodaltons. And in fact, we saw that they were several that were running at the correct size, which is encouraging and not a lot of degradation was seen. So since we saw that it was encouraging, we then took the soups and used affinity chromatography to pull out the GB protein to purify it from the soup. After we isolated the proteins, we concentrated the protein down to do a stability test. If the protein was unstable, we were going to see precipitation as the protein crashed out into pellets. So after we completed the stability test, we did have precipitate, meaning that it was unstable. So we ran a gel for both the pellet and the soup side by side. So we saw that quite a few of them were a bit unstable, but not completely unstable because we can see a lot of protein ended up in the soup, meaning that they are soluble and at the correct size. And here, for example, under C2, you can see that there's like 50-50 in the soup and in the pellet. And then after this, I won't be showing the results, but we did also do a binding test that showed that these mutants all bind to an antibody that only binds to prefusion GB, which is a hopeful sign that these all are folded correctly in prefusion form, which is our goal. And this is very encouraging. And our next step is to stabilize further so that they do not crash out. And to do that, the next step is to introduce a stabilized mutation at the tail. So right with this red circle is what we think will increase the stability of the GB, of the longer GB. And for my reflection, I just want to begin to say that I really entered this internship with no idea of what to expect. It was very fast paced, but very engaging. I really appreciate working in the lab. It just gave me an experience that I want to continue on as I continue on to my studies. I don't want to be, I am aiming to be a nurse, but my major is public health. So I want to just keep my options open and to definitely like bring awareness to vaccines because CMV is actually not very well known or discussed. And I am happily and proudly able to say that I will be leaving this lab with knowledge and efficient in knowing how to do medi-preps, read protocols, and the most important struggle that I had through my internship was doing math and the conversions. So I am very grateful for that. And I want to go ahead and say I'm very thankful to my mentor, Javer Najib. And I also want to go ahead and give a big shout out to Anthony Buzanko for taking the time to educate me and push me. Both of them pushed me with each experiment we did. I also want to thank Professor Dudetsky for allowing me to take part in his lab. And I also want to thank everyone in the lab for being very kind and encouraging and helpful and a helpful resource throughout my internship experience. I also want to thank Dr. Amy Edwards, as she was the one who inspired me to continue pursuing a career in science. And lastly, I want to thank Sophia Kim, Amanda Carvajal, and the whole SLI team for just allowing me and choosing me to be a part of this summer internship. Thank you.
Thank you, Mishelle. That was wonderful.